So my project was on the impact of the S allele of the gene SLC6A4 on depressive disorders. And my mentor was Professor Amanda Storm from Western Carolina University. So let me just, okay. So here's my agenda. I split into 6 sections, just like my research paper: an introduction, a review of the S and L alleles, a review of recent studies, role of other factors in this field, current challenges, and the conclusion. My research paper was in the literature review format, so yeah. So first we're gonna do the introduction to the SLC6A4 gene and the SERT protein. So first, I'm going to talk about the neurotransmitter serotonin, which is a neurotransmitter chemical produced from tryptophan in the brain, and it's commonly known as the feel-good chemical. And it's responsible for a lot of things such as the regulation of mood, sleep, cognition, etc. And then we're going to talk about the serotonin reuptake transporter protein, also known as SERT. That's what I'm gonna call it for the rest of the presentation. It's the protein that is used for the purpose of transporting serotonin from synaptic spaces to presynaptic neurons and ending the serotonin cycle. It's a big target for antidepressants because it's really, really crucial in the cycle of serotonin in the brain. And when the SERT protein is dysfunctional, it has a very, very big correlation with psychological disorders and especially depression, which is what I'm studying. The gene SLC6A4 is the gene that encodes for the SERT protein, and it's located in the chromosome 17 in the human genome. And there are many names for it, such as 5-HTT SERT and 5-HTT LPR, which you may see later in the presentation. And this gene has a polymorphism, which is basically when a gene has 2 different versions or more. And we will talk about that right now. And here's a diagram of the physiology of the serotonin cycle. And here serotonin is labeled as 5-HT, and the SERT protein is labeled as a 5-HT reuptake transporter. So you can see that tryptophan is turned into serotonin, and then it gets released into the synaptic cleft by the presynaptic neuron. And then it is received by the 5-HT receptor on the postsynaptic neuron, and then it is released back. And basically, SERT's job is to take this serotonin back to where it came from and terminate the cycle. And then now we're going to talk about the S and L variants of SERT. So as I said earlier, polymorphisms are when 2 plus possible variants or alleles of a gene exist within a population. And the specific polymorphism that I'm studying within the SLC6A4 gene is called 5-HTT-LPR. And then within this polymorphism, there are 2 main alleles that are mostly studied. The S allele, the short, low expressing allele, and the L allele, which is long and high expressing. So the difference between these two is basically just the length. The L allele has a little bit more encoding than the S allele. Each human, because they inherit 1 allele from each parent, they have 3 possible combinations that they could have. The L and L, which is dominant, and the S and L, and the SS, which is recessive. And here is another photo to better explain the gene structure. You can see that it's in the human chromosome 17, and In this region, the promoter region, which is basically responsible for the transcription of the SERT protein, there are two different alleles in the polymorphism. And you can see like the difference in length and the amount of mRNA they produce. And now I'm going to go over a review of recent studies which researched the impact of the S allele specifically on the risk of depression and other depressive disorders. The first study that I'm going to go over is by Dr. Daniel Weinberger and his team at the National Institute of Mental Health, who studied neurotypical individuals. And they found that people with recessive allele combos had amygdala sizes 25% smaller than individuals with dominant allele combo. And the amygdala is the part of the human being that's responsible for emotions, especially fear. A smaller amygdala leads to smaller regulation or lesser regulation, rather, of fear responses. Because of the lesser fear regulation, people with recessive combos may be prone to depressive disorders, as being in a continuous state of fear really worsens your mental health. The next study that I'm going to discuss is by Dr. Klaus Peter Lesch and his research group in Germany. They did a study where they examined healthy individuals and their responses to making mistakes in video games, and they were all rigged basically, so there was no way to win, and the prize was a cash bonus. And they found that people with the SS combo have a bigger response to making a mistake. And more specifically, they have a bigger electrical event that occurs in the center of the frontal cortex, which is also responsible for managing emotions. So this implies that these carriers tend to have more negative views and beliefs regarding life events. But it should be noted that they only concluded that they have a negative view of life events, but it doesn't really say much about other factors such as like childhood events that affect their worldview. In another study done by Claudia Delicali and her team in 2022, the interaction of the 5-HTT-LPR polymorphism and stress also is influenced largely by time. So what I mean by that is that when a person experiences chronic stress, which is long-term, depression risk is much higher compared to people who only experience acute stress or stress for a short period of time. And then finally, we're going to talk about a study by the Baltimore Epidemiologic Catchment Area in 2020, which used a population sample evaluated with the schedules for clinical assessment in neuropsychiatry by qualified psychologists in order to investigate the correlation between the polymorphism and the psychiatric morbidity and comorbidity of certain disorders. And what that means basically is that which disorders have a higher association with each other. And I will be showing the results on the next few slides, which will make more sense. So here's table one. You can see that there's a carrier to non-carrier ratio. So by carrier, they mean people who carry the S allele, and by non-carrier, they mean people who only carry the L allele or dominant allele combo. And you can see that being more than one shows that the percentage of S carriers within psychological disorders outweigh the percentage of non-S carriers. And it can be seen that major depressive disorder, also known as MDD, has a lack of association within carrying the S allele for the polymorphism. And other depressive disorders have similar ratios that aren't necessarily significant. And this shows the comorbidities or the associations between different depressive disorders. And actually, very interestingly, people with major depressive disorders who had the S allele were 5.1 times more likely to have a social phobia than non-carriers. And these individuals were also 2.3 times more likely to have agoraphobia. So there are different factors that influence depression, and we will talk about that right now. Epigenetics is the study of how the environment influences the expression of genes. And there are different epigenetic processes that happen within the body, such as non-coding RNA, post-translational histone modification, and DNA methylation. And these can influence how certain proteins are produced by the polymorphism in the gene. And also, there is severe trauma, which is not properly measured in certain studies. For example, the Baltimore study, that they had a disclaimer that they did not account for previous trauma or life events that had occurred to the individuals. Another factor is early life stress, which is basically stress experienced in the early stages of life, and prenatal stress. In a study done on rodents, they measured that rodents with wait, sorry, rodents and early life stress, like baby rodents, they had interactions with um, the SLC6A4 gene and the hypothalamic pituitary adrenal axis. And prenatal stress also plays a role. For example, disrupting mother pup interactions in rodents also affects the regulations of the HPA axis and also changes the neurochemical functions of an organism. And the main takeaway is that a disorder as complex as depression is not necessarily determined by just one gene or just one polymorphism. And here are the challenges in the field and in the research done. As you can see earlier, many studies don't show conclusive results, and there isn't a clear line drawn between S carriers and L carriers. And there are also new findings on CERT and the gene itself that could change everything we understand. For example, mediated CERT expression control, also known as MIR16, it's a non-coding RNA molecule that binds to the mRNA of the gene, and that reduces the amount of CERT protein produced. So that is not considered in many studies as of yet, and it might also have an effect on depressive disorders. And a third polymorphism in the SLC6A4 gene has been discovered, which not only opposes the idea of CERT being biallelic, which means having two alleles that have been studied extensively, the S and L alleles, it also changes how we view the polymorphism. And it could be that we've been studying this polymorphism wrong the entire time. And here's my conclusion. As you can see, the SLC6A4 gene encodes for the CERT protein, and it has a polymorphism, which is believed to be biallelic. And many studies investigated the link between the S allele of this polymorphism and many depressive disorders. There had been overall there has been a potential correlation that has been found, and still there are many factors not considered, such as the environment, epigenetic processes, trauma, etc. And also there are new findings that are not considered as well, such as the third allele in MIR16. Okay, well, that's the end of my presentation. I'd like to sincerely thank Professor Amanda Storm for being my mentor and guiding me through the whole process of creating a research paper, especially when I have not done anything like this before, and the fact that we live in two different states. I'd also like to thank Talaria for even letting me into this program for free and letting me delve into research for the first time in my life. Yeah, thank you.